So this is a quick start guide for BMDExpress 2.0. I've already run the analysis and the data tree is populated, but it would obviously be empty if you were just starting off. So the first thing that you're going to have to do when you open BMDExpress is you're going to have to update the annotations. And so you go to File, Update Annotations, and this will populate with all the platforms that are supported by BMDExpress. You will pick your platform, say it was the Affymetrix Rat Genome 232.0 array, select that and download it. We have support in BMDExpress for BioSpider platforms, Agilent platforms, Illumina platforms, and then if you have performed RNA-seq, we have RefSeq and Ensemble IDs, annotations, and we cover 5 species: rat, Drosophila, human, mouse, and zebrafish. So once you select your platform, just hit update, and then once it's finished, hit done. After that, you're going to have to load your data. Your data should look like this in a tab delimited text format. So this is a tab delimited text file loaded into Excel. And what you can see going down the side here are the probes. This happens to be the S1500 platform, but it could be Affymetrix microarray. So if this is the 232.0 rat array, this would be about 31,000 rows. Across the top are the sample IDs, and then just below that are the doses, and 0 dose, and then the next dose. They can be out of order if that is the case. The software will recognize it and orient it correctly. Then, of course, in the matrix is the data. Typically, this is log2 transform normalized data. BMDExpress does not perform normalization, so you'll have to normalize and format these files outside of BMDExpress. Once you've done that, you can then load them into BMDExpress. So you go up here to File, Import Expression Data. And you go to the folder that contains your data, and this is just example data. And if you want to batch load them, you shift click and hit open. If you just want to load one, you select that one and hit open. And what it will do, we'll just load one now as an example. If you have downloaded the annotation for your platform, it should recognize the platform by matching up the probes. If it does not, you'll just have to scroll down through this drop-down menu and find your platform, and then hit OK. And it will show up over here. So we're just going to remove this now to avoid confusion, so you can delete stuff out of the data tree if you so need to. The next step that you're going to want to take is to filter your data to remove probes that are not responding to chemical treatment. And so you go to Tools and perform one-way ANOVA. And so here I've multi-selected, shift-click under expression data, then went to one-way ANOVA, and you have a bunch of parameters that you can set to perform the one-way ANOVA. The p-value, whether or not you want to perform multiple testing correction. If you have an Affymetrix platform, you can remove the control probes that have an AFFX prefix. And then you can choose a fold change cutoff to remove genes that don't change within a certain range. And then you have to tell the software whether or not your data is log transformed. Most data that's normalized for gene expression are log transformed. And so if the data is log transformed, leave this checked. If it is not log transformed, uncheck it. Also, you will need to tell the software what base log you have transformed the data with. Once that's done, you hit Start, and it'll start running the ANOVA. And then when it's done, it will populate here. And so I've run these previously, so I've run all 3 data sets. And you can see the names are actually quite informative. It tells you one-way ANOVA, you use the P of 0.05, whether or not you use a multiple testing correction, and whether you use a fold change filter or not, and what that fold change filter was. So when you get to this, you click on one of the one-way ANOVAs, it populates as follows. So you have your parameters that were associated with your one-way ANOVA, your results, which include if your data was matched up to a platform, it will contain the probe ID, and then the gene symbol, and the entree gene IDs, and then the gene symbols, and then a number of statistical parameters, and then fold change values, and then fold change values at each dose level. Above that are a number of visualizations. There are your traditional volcano plots. There are also a number of other plots that you can look at that are related to the results in the table. If you need to, you can hide the table. You can hide the filter. So the filter function is what you can do is you can actually set certain standards. So if you wanted to change your max fold change to greater than or equal to 3, you'll notice when I put this in the put this in the box there, it auto updates both the table and the volcano plots. And if it's not doing that, you have to hit the apply filter radial button here. Once you apply the filter, it will start. It will then adapt to the filter selection. So after you've run the one-way ANOVA, you will want to then perform a benchmark dose analysis. This is where the data is fit to multiple curves provided from the EPA's BMDS software. And so what I've done is I've shift-clicked it and selected all three to run all three at once, or you can just select one and go to Tools and Benchmark Dose Analysis. And then you select which models you want to run. We've incorporated exponential models now. The older version did not have that. And then a number of parameters, the model selections process. And then if you have multiple threads, we recommend you use them because the modeling process can be computationally intensive. So you can just change the number down here and then select Start. And all of these parameters are described in the documentation, and you have the options of changing them if you so choose. You hit Start, and they run. It's going to take a while to run in certain cases. If you have a large number of data sets and a limited number of threads, it can take a significant amount of time. So once you hit Start and it runs through, it'll populate here under Benchmark Dose Analysis. And this is a benchmark dose analysis result from the combination of the ANOVA and example data set 1. And so when you first see this populated, you select the benchmark dose analysis for example data 1. You have on this lower pane here all the variables associated with your analysis, the model types, the date stamps, things along those lines. And then you have over here the data table for all the BMD results for each probe. And if you want to see an individual probe and its fit, you can select right there. And you have, so basically what I did there was I was in the table and I selected one of these probes, and it shows up as the data is plotted with the best fit model, but you can change the model if you so choose by dropping down here. You can also log the axes if the spread on your doses is very wide, and you can perform mean and standard deviation plotting. And in addition, there's the BMDL, BMD, and BMDU values, the fit p-value, and the Iacocki score. You can change the properties if you so choose. By going into the Properties button, you can change the axes under Domain Axis and Range Axis by clicking on the Range button, or the Range tab in the Domain Axis, or the Range tab in the Range Axis. So, and also, if you want to click through the table, these plots are interactive, so you can have this plot open in the front, and you can just click through the table if you so choose. You can also look through the table here, so you can change the probe here also. Just like with the one-way ANOVA, there are a number of filters that you can set, and you can add remove filters by clicking add remove filters. So if there's something that you that's in the table that you would like to filter by, you can select it and then hit done, and it'll populate over here, and then you can use the drop-down menus to select and then set your values. In the visualization area, we first have a pie chart that shows the distribution of best fit models. So in this case, 27% of the best fit models are Hill models, 6.8% are linear. We have a best BMD to BMDL scatter plot. You should see roughly a 1-to-1 relationship here. If you don't, that usually means your fits aren't so great. Then we have histogram distributions of the best BMD, and you have a number of other visualizations that you can look at here. Just like in the ANOVA, you can hide the charts if you want to, you can hide the table if you want to, and you can hide the filter if you want to. In addition, you can come up here to the benchmark dose analyses, and we have a more detailed view of the parameters, so you can right-click and say select hill, and you have additional parameters on the far end for each hill model if you want to look at them. Same is true with all the model types. So once you're done with your benchmark dose analysis, you're going to want to perform a functional classification. So what you do is you go under your benchmark dose results, and you can shift click and click multiple if you want to do them in batch. If you want to just do one, you select one. And then what you go up to here is you go to Tools, and then you go to, say you want to do a gene ontology functional classification. These are provided with the software, so you don't have to provide the files. If your platform was identified by the software, then this should work. And so you come in here to you select the gene ontology, and you can select either a universal or a subset of the Go biological processes. You then determine whether or not you want to remove certain probe sets based upon certain parameters. These are all described in the documentation. And then you hit start, and what will happen is it'll take the probes that have been fit to curves here with certain standards that you've set, and we'll pass them into the Go biological processes. And then from there it will take those and determine a number of benchmark dose values, median values, mean values, and BMDL and BMDU values. And so that's what's contained in this file here under functional classifications. It's actually the human Go biological process, and it tells you that you've removed promiscuous probes, and you've also removed BMDs greater than the highest dose from the category descriptive statistics. That's what the true means there. So very informative names associated with the files. So in the table, you have all the Go biological processes that were populated. And what you're probably going to want to do is, what common practice is actually determine which Go biological processes are actually reasonably populated, and then report the BMDs associated with those gene sets. And so what we do is we first select apply filters, and then we select what we want to do. And in most cases, we use a percentage. So a Go term has to be X% populated, in this case 5%. And there has to be at least 3 genes in order to call a median BMD out of that category. So both of these have to be true in order for something to show, for a Go term to show up here, or in the case of signaling pathway analysis, that would have to be true also. So once you have run this, you will see this in the table, and you can go down to the table here, and you can sort the table by most potent BMD median or most potent BMD mean. And if you need to, you can move the columns around, so you can grab a column and move it to the other side of another column, so you could stack them if you need to visualize them for some purpose. And then you can, if you need to, you can further modify the filters, and you'll notice that both the plots and the corresponding data will change when I change this number, so it filters on the fly. One note is when we go to export this data, the filters do not apply, so everything pre-filter is exported. So you'll have to filter it again in Excel or some other tool to get back what you have in the filtered table here. So in the plots we have for the benchmark for the functional classifications, first we have this what we call an accumulation plot. So what we do here is we take all of the Go terms in this case, and we rank them by their median BMD, and we provide and we associate that rank with a number. So say in this case what we have is the lowest BMD median is associated with positive regulation of embryonic development, and you'll see it's given a value of 1, or an accumulation value of 1. And as you mouse up here, you see that there are 2 additional Go biological processes, and that's given a score of 3. And as you keep moving up here, this is a Go biological process that's associated with a large number of Go terms, all of which have the same BMD median value, and this one has an accumulation of 800. So as you go up, the rank goes up, and so we use this to visualize potency. Then there is the bubble plot. The bubble plot is simply the BMD median going from lowest to highest against the log of the Fisher's exact two-tailed test for the enrichment of the Go biological processes, and the size of the bubble is the percentage populated, in this case, the Go term is. If you mouse over these, the Go term will pop up, or the pathway name if this is a signaling pathway analysis. We also have the BMD median to BMDL median, and we have all the different mean, median, and then ratios to BMDU to BMD, various other sundry scatter plots, all within the drop-down menu underneath, just above the visualization box. But as you see here, what we have plotted here is a BMD median to a BMDL median, and you should see roughly a 1-to-1 relationship if your data is fit reasonably well. You have other drop-down, other features here. You have a box and whisker plot, which shows you, say in this case, we can open this up. This is Go term 212, and it shows you the BMDL, the BMD, and the BMDU for that Go term. Then you can look at a number of other, the BMD and BMDL bar charts. These are simply looking at the BMD values plotted against the Go terms on a bar chart. There are additional accumulation plots if you want to look at the BMD median, BMD mean, BMDL median. So we have all of these plotted as accumulation plots. And again, these are all filtered based upon the filter standards that you've applied and whether or not you press the Apply Filter button. Best model pie charts, very similar to what was in the benchmark dose analysis result up here when you select that. The mean histogram, this is just a distribution or a count versus the BMD median. And so in this case, it looks like the peak BMD median was right around 200. And then we have the BMDL mean histogram and a number of other histograms, the median histograms, and then more scatter plots, all for visualizing the data. So you should be careful. Sometimes there are more plots below the plots that you can see, so you can hide the table and actually take a look at those. Finally, there is a functionality in viewing whether you're viewing data in the one-way ANOVA or the benchmark dose or in the functional classification. What you can do is you can add additional data sets. So I will add the 2 other Go biological process analyses, and they will co-plot for a number of different visualizations. So as you can see here, we have example data 1, example data 2, and example data 3 all in the accumulation plots. This populates, this type of viewing can be done in the bubble plots, in the BMDL versus BMD scatter plots, and also in the box and whisker plot. So what's nice about the box and whisker plot too is you can, if you wish to, you can simply filter for 1 or 2. So you can shift click or control click certain Go terms, and this is true in the benchmark dose analysis and then also in the one-way ANOVA, and you can click draw select items, at which point it only shows you the items that you have selected in the table, and it'll show you across those that are populated in the add remove data set and visualization. So if you wanted to get rid of this or get rid of some of the data sets, you just go into, you press minus, and this pops up, and then you hit done after you've selected them, and now they're gone. And so you can go back to the visualization. As you can see, all I have is 2 selected, so there's only 2 dots here in the accumulation plot, and there's only 2 dots here in the bubble plot. And you can always shift click and add more if you so desire to. And again, if you want to add more data sets, you just go back there and add more, and there they are. Some other nice features of the software include what's called a spreadsheet view. So if you want to look at an individual data set and compare it to another data set, what you can do is you can select multiple data sets and open them in spreadsheet view. And so what we have here is example data set 2, shown over here, and example data set 1 over here. And if you wish to overlay or filter them differently, you can match them up. You can hide the table and hide the charts if you would, or hide the filter so you can get a better view. Do the comparisons. You can add samples if you so desire, or you can compare them. So this is another nice feature of the software. Well, best of luck in your BMDExpress analysis, and if you have any questions, you can contact me at auerbach@niehs.nih.gov, and I hope I can help you along and make your research, facilitate your research and make it successful.